Hello, everyone. Our names are Tate Bertea,
And Ryan Shaffer.
and for our community project assignment, we have decided to take a look at the stories of the discovery and mechanisms behind several major types of muscular dystrophies.
We chose to research this topic because we really only get a major view of Duchenne muscular dystrophy in PhySci 121, and we are interested in seeing the mechanism behind other forms of muscular dystrophies.
We will begin with a brief review of Duchenne muscular dystrophy, and then we will take a look at several other types of muscular dystrophy.
Duchenne muscular dystrophy is a disease which is characterized by weakness in the muscles of affected individuals, learning disabilities, and loss of ambulation around age 10 to 12, and early mortality between ages 20 to 30. Duchenne muscular dystrophy was first described by Guillaume Benjamin Amand Duchenne in 1861. He was the first to do a biopsy of tissue from a living patient for microscopic examination, and he used these biopsies to study the effect of Duchenne muscular dystrophy on muscle physiology. It wasn't until 1987, however, that the cause of Duchenne muscular dystrophy was elucidated through the use of human genomic analyses. The disease was determined to be caused by the lack of a functional protein or the lack of functional protein called dystrophin. Dystrophin is an important component in a protein complex which anchors the sarcoplasm to the extracellular matrix. The lack of a functional copy of dystrophin causes the muscle cells to become much more fragile and they become very easily damaged as well. Duchenne muscular dystrophy has been observed to disproportionately affect males. Roughly 1 in 3,500 males are affected. This is because Duchenne muscular dystrophy is an X-linked recessive disease. This means that males only need one copy of the mutant dystrophin gene, while females need two in order to inherit the disease. Despite this, female carriers of Duchenne often experience cardiomyopathy, a weakening of the heart, as a result of having only one healthy copy of dystrophin.
Becker muscular dystrophy was first described by Peter Emil Becker in 1955. It was noted that Becker muscular dystrophy causes a very similar yet milder phenotype to Duchenne muscular dystrophy. The symptoms of Becker muscular dystrophy typically do not manifest until after about 10 years of age and may not appear until much later on. Similar to Duchenne muscular dystrophy, it was found that Becker muscular dystrophy is caused by mutations involving the dystrophin gene. The wide range in the severity and the onset of Becker muscular dystrophy can be attributed to the varying degrees of severity of the different mutations which can cause the disease. Becker muscular dystrophy is caused by mutations to the dystrophin gene which do not severely alter the function of the dystrophin gene. Typically, the mutations which cause Becker muscular dystrophy are deletions in portions of the protein which are non-essential for proper function. An important difference between the deletion mutations, which can cause Duchenne, and those that can cause Becker, is that the reading frame of the functional dystrophin protein is maintained. This means that even the components of the protein which come after the mutation are still properly synthesized, which is the reason that dystrophin in Becker muscular dystrophy patients is still partially functional.
Limb-girdle muscular dystrophy is not one type of muscular dystrophy, but rather a large group of disorders with many subtypes. The common symptoms of all limb-girdle muscular dystrophies are atrophy and weakness in the proximal muscles of the hips and shoulders. Limb-girdle muscular dystrophy was first described by John Walton in 1954. He defined it as separate from other forms of muscular dystrophies like Duchenne and Becker because it was not observed to be inherited in an X-linked fashion. Today, over 30 distinct types of limb-girdle muscular dystrophies have been identified. Limb-girdle muscular dystrophy results from mutations which cause at least one of the proteins known as the sarcoglycans to be non-functional. Like dystrophin, the sarcoglycans are an important component of the protein complex which anchors muscle cells to the extracellular matrix. The absence of even one of these proteins can cause increased muscle damage and weakness. As mentioned earlier, limb-girdle muscular dystrophy is not an X-linked recessive trait. It is actually inherited autosomally, and the different subtypes can be inherited either dominantly or recessively, and the nomenclature for each individual subtype of the disease is dependent on the type of inheritance. Limb-girdle muscular dystrophies, which are inherited in an autosomal dominant fashion, are called LGMD1s, and limb-girdle muscular dystrophies, which are inherited in an autosomal recessive fashion, are called LGMD2s. There are more LGMD2s than there are LGMD1s, along with a greater frequency of LGMD2s.
Fascioscapulohumeral muscular dystrophy, also termed FSHD, is typically characterized by weakness and atrophy in the muscles in the face, shoulders, and upper arm. This is where the term fascioscapulohumeral comes from, because facies is the medical term for the face, scapula is the medical term for the shoulder, and humerus is the anatomical term for the bone which extends from the shoulder to the elbow. FSHD was first described by Louis Landouzy and Joseph Dejerin in 1884. They differentiated it from Duchenne muscular dystrophy because of its characteristic effect on facial muscles. The two also noted the disease was passed down genetically after observing its presence in 4 generations of a family. FSHD can be divided into 2 categories: adult onset and infantile onset, depending on when the symptoms begin to appear. FSHD is inherited in an autosomal dominant pattern. The disease is associated with mutations which cause inappropriate expression of the DUX4 gene. In individuals without FSHD, DUX4 is only expressed early in human development and repressed in mature tissue, as seen in the control. However, in individuals with FSH, this repression of DUX4 is compromised by mutations in regions surrounding the gene, ultimately translating DUX4 protein. It is currently unclear how exactly the expression of DUX4 causes muscle damage, but it is known that the DUX4 protein influences the expression of hundreds of other genes. Some of the genes which are influenced are involved in muscle function. Research has also suggested that the presence of DUX4 in cells leads to the production of potentially toxic proteins.